Hello friends, in the previous video we discussed about initiation step of eukaryotic DNA replication. You can refer my video, the link is given in the description box. In this video we are going to discuss about elongation step of eukaryotic DNA replication. Elongation is the synthesis of two new daughter strands complementary to the template strands. It is the second step of eukaryotic DNA replication. Now, what is elongation or what happens during elongation? Let us see the overview or let us summarize it. Range of proteins bound during initiation, and these then unwound DNA at the origin. So, as we have seen in the initiation step, that number of proteins comes and bind to the origin of replication. This results in the unwinding of the DNA. This takes place in initiation. Then what happens? DNA polymerases are loaded. These then extend daughter strands with the help of certain other proteins. So in this video, in the elongation step, we will discuss about these in detail, that how the polymerases are loaded and how the extension of the daughter strands takes place, or how the elongation occurs, and what are the other proteins that are involved in this step. Some of the facts of DNA elongation. Direction of elongation, or the direction in which the new strand is synthesized, is always in the 5' to 3' direction. Primer is required. Major replicative or the main replicative enzyme that are involved in the elongation cannot initiate the synthesis of new strand. They always require 3'OH group for the extension or elongation of the new strand. Hence, primer is required. Addition of new nucleotides require energy. So, in order to extend or elongate the new strand, nucleotides are added which are complementary to the template strand. And during the addition of nucleotides, energy is required in the form of ATP. Elongation is semi-discontinuous, as DNA is antiparallel, and the elongation or the synthesis of new strand always takes place in the 5' to 3' direction, and hence one of the daughter strand is synthesized continuously, which is known as leading strand, and the other strand is synthesized discontinuously, known as the lagging strand. Hence, DNA replication is semi-discontinuous in nature. Proofreading also occurs in elongation. Sometimes wrong base is incorporated. DNA polymerase, when recognize this wrong base, then it moves backward, remove or excise this base, and this is followed by the reinsertion of correct base. This is known as proofreading. Now let us discuss enzymes and proteins involved in elongation of eukaryotic DNA replication. First is DNA polymerase alpha. This DNA polymerase alpha synthesizes RNA primer, followed by the synthesis of a stretch of DNA. Primer is synthesized once on the leading strand and at the start of each Okazaki fragment on lagging strand. DNA polymerase delta. It is responsible for the synthesis of lagging strand. It is highly processive due to the sliding clamp pCNA. It has 3'5' exonuclease activity, hence it shows proofreading activity. Now DNA polymerase epsilon, it synthesizes leading strand. It also has high processivity due to pCNA. It also shows 3'5' exonuclease activity, or it has proofreading activity. Then CTFO. This is required for the recruitment of DNA polymerase alpha. pCNA, proliferating cell nuclear antigen, which is sliding clamp. It increases the processivity of DNA polymerase. RFC, it is clamp loader, it loads pCNA. It is responsible for polymerase switching, that is replacement of DNA polymerase alpha by main replicative DNA polymerase, that is DNA polymerase epsilon and DNA polymerase delta. Chain elongation reaction, it is same as prokaryotes. So as the nucleotides are added during the synthesis of the new strand or the extension of the new strands, then certain reaction takes place. It is same as in the prokaryotes. This we have already discussed in elongation step of the prokaryotic DNA replication in detail. You can refer my video, the link is given in the description box. Now to understand the elongation step, let us start with some of the events that takes place during the initiation. This we have also discussed in the previous video. So we have seen that during the initiation, when CMG complex is formed, the CMG complex is functional replicative helicase. Now what happens? Parental DNA unwinds at the origin, that is the two strands of the DNA separates. This results in single strand of DNA at the origin. Now the MCM10 is recruited or it attaches to the CMG complex. This MCM10 then facilitates the recruitment of RPA, replication protein A, which is single strand binding protein which binds to the single strand of the DNA and stabilizes it, and also facilitate the recruitment of DNA polymerase alpha with the help of CTF4 protein. DNA polymerase alpha then synthesize RNA primer followed by a stretch of DNA. So what happens? CMG complex is formed, which consists of CDC45, MCM227 and GINS. This is functional replicative helicase. This causes the unwinding of the DNA at the origin. Now the two strands of the DNA unwinds and this results in the single strand of the DNA. MCM10 attaches to the CMG complex. It facilitates the recruitment of RPA, which is single strand binding protein, and it stabilizes the single strand of the DNA, which results due to the unwinding of the DNA. Also, DNA polymerase alpha is recruited. This DNA polymerase alpha synthesizes stretch of RNA primer, which is in orange color, followed by synthesis of a stretch of DNA, which is in green color. Then RFC, replication factor C, which is clamp loader, causes dissociation of DNA polymerase alpha. Because DNA polymerase alpha is not major replicative polymerase as it has low processivity and it also lacks the 3-5- exonuclease activity. Hence, the DNA strand that is synthesized by DNA polymerase alpha is not accurate. And after the dissociation of this DNA polymerase alpha, this RFC also causes the assembly of pCNA, which is proliferating cell nuclear antigen, which is sliding clamp, same as the beta clamp, which holds DNA polymerase delta and DNA polymerase epsilon to the template strand when loaded. So this is responsible for the processivity of DNA polymerase delta and DNA polymerase epsilon, which are the major replicative DNA polymerase. So this process in which DNA polymerase alpha is replaced by the major replicative polymerase, DNA polymerase delta and DNA polymerase epsilon, is known as polymerase switching. Then the DNA polymerase epsilon synthesize leading strand. Also DNA polymerase delta synthesize lagging strand. DNA polymerase epsilon is already recruited to the CMG complex, but it cannot initiate the DNA synthesis as it requires 3-OH group for the extension of the new strand, which is provided by the primer. Hence, once the DNA polymerase alpha synthesizes the primer, then only the main replicative enzyme DNA polymerase epsilon and also DNA polymerase delta is loaded. Primer is synthesized by DNA polymerase alpha once for the leading strand and at the start of each Okazaki fragment for the lagging strand. DNA polymerase alpha, because of its slow processivity, and it also lacks the 3-5- exonuclease activity. Hence, the DNA that is synthesized cannot be accurate. It is not the main replicative enzyme. So, once the primer is synthesized, this DNA polymerase alpha is dissociated and the main replicative DNA polymerase, that is DNA polymerase epsilon and DNA polymerase delta is loaded. So what happens, this RFC, which is replication factor C, which is also the clamp loader, causes the dissociation of the DNA polymerase alpha and assembly of pCNA, both to the template of the lagging strand and to the template of the leading strand. This pCNA holds DNA polymerase epsilon and DNA polymerase delta, and it is responsible for the high processivity of this main replicative DNA polymerase. So the process of replacement of DNA polymerase alpha by the main replicative DNA polymerase, that is DNA polymerase epsilon and DNA polymerase delta, is known as polymerase switching. So this DNA polymerase epsilon synthesizes the leading strand, DNA polymerase delta synthesizes the lagging strand. This lagging strand is synthesized in the form of the small fragment. So DNA polymerase delta synthesizes a stretch of the DNA, then it gets dissociated. Then it is again loaded to the next primer, and the process is repeated again and again till the completion of the synthesis of lagging strand. So what we have discussed, that one of the strand is synthesized continuously and the other strand is synthesized discontinuously in the form of the Okazaki fragment. For the synthesis of this continuous strand, known as the leading strand, DNA polymerase epsilon is loaded once and then it continuously synthesizes the new strand. But for the synthesis of the lagging strand, DNA polymerase delta is loaded, then it synthesizes a small stretch of the DNA. Then it gets dissociated and is loaded to the next primer. Then the process is repeated. This process is repeated again and again till the completion of the synthesis of the new strand. So why this is synthesized continuously and this is synthesized discontinuously? This is because the new strand is synthesized always in the direction of 5' to 3'. Since this strand, the 5' to 3' is in this direction, and hence it is synthesized in the direction same as the movement of the replication fork. As the replication fork moves in this direction, the double helix of the DNA separates, and hence the single strand template for the synthesis of this strand is continuously available. Hence it is synthesized continuously. But if we talk about this strand, here 5' to 3' is in this direction, which is opposite to the direction of the movement of the replication fork. As a result of which, small stretch of the single strand template is available, small stretch of the DNA is synthesized. Then as the replication fork moves or the double helix of the DNA separates, again a small stretch of the single strand template is available. Then again small stretch of the new strand is synthesized. In this way, this process is repeated again and again till the completion of the synthesis of new strands. Hence, it is synthesized in the form of the small fragments known as the Okazaki fragment. Also, this strand is known as the leading strand and this strand is known as the lagging strand, because synthesis of this strand starts early, and when a certain stretch of this strand is synthesized, then only a stretch of single-stranded template is available for the synthesis of this strand. Hence, the synthesis of this strand starts late compared to this strand. So, this strand lags behind this strand. So, it is known as the leading strand and it is known as the lagging strand. This is all for today's video. In the next video, we will discuss about primer removal and its replacement by DNA. So, stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.